sodiums. At that point, it becomes a different shape so that now potassiums can bind. And the binding of the potassium kicks off that phosphate, changes the shape back to the original shape that would fit sodium, and spits potassium into the cell. So notice we have a high concentration of sodium outside the cell, and what direction are we moving the sodium? To the outside of the cell. We have a high concentration of potassium inside the cell. What direction are we moving the potassium? Towards the inside of the cell. So we're building a concentration gradient. Bulk transport also requires a lot of energy. It is actually reshaping the cell membrane. So you reshape the cell membrane to either surround something outside the cell and bring it into the cell, or to let one of those vesicles that's moving around the cell melt into the cell membrane and release stuff to the outside. So doing that kind of reorganization of the cell membrane does take energy. So when you're releasing stuff from the cell, that's called exocytosis. You're exiting. When you're bringing things into the cell, that's endocytosis. Endo means inside. And there are 3 different types of endocytosis. The first 2 just differ based on what you're taking in. If you're taking in a solid chunk of something, it's called phagocytosis. In fact, there are cells in your immune system that are called phagocytes because they're white blood cells that go around and eat either dying cells or cellular debris, or they eat pathogens. Penocytosis you don't tend to hear about very much. That's cell drinking is literally what it means. You basically are taking in liquid or a bunch of small particles dissolved in it. And then receptor-mediated endocytosis, you have receptors on the surface. When a specific molecule binds, that's going to trigger it to endocytose. All right, so here are pictures of each of them. So this is phagocytosis, where you are taking in a solid particle. Penocytosis, where you are just taking in some sort of fluid or something dissolved. And receptor-mediated endocytosis, you have receptors. Once something binds, that triggers them to come in. If you go into higher levels of biology, you'll learn the purpose of those, but we're not going to get into that. Exocytosis is the opposite process. Like I said, you have a vesicle come up. It basically melts into the membrane and spits out its contents. Then that membrane that was part of the vesicle is now part of the cell membrane. You do have electrogenic pumps. You can also have co-transporters. So proton pump uses energy to build a proton gradient. A co-transporter or a symporter will usually use a concentration gradient of one thing to move something else that would otherwise require energy. So it's a way to make your active transport passive. But then you usually have to build that concentration gradient in the first place. All right, this is just a GIF, if it works, showing plasmolysis. So these are cells of something called Elodea, which is a plant that's found in water, and it doesn't look like it's working, which is weird because it worked when I did this at home. What ends up happening is the cell membrane pulls away. You can actually see that. Let's go to water potential instead. So this should be a familiar diagram. You've seen it since freshman year. The hypotonic, isotonic, hypertonic solutions. You can think of this in terms of the potential energy of water, or the water potential. It's how much energy the water has to do work. So how free are the water molecules, and are they free enough to do work? So if you have a salt crystal dissolved in water, in the beginning, before any of the salt ions have moved out into the water, you'll have a lot of free water. It's free to do work. But as those start breaking off from the salt crystal and start interacting with water, notice this water, when it's around an ion, is no longer free to do any work. It's stuck around that solute. And as more and more of the water ends up being stuck to solute, less and less of it is able to do work. So as you add solute to water, it will decrease the water potential. We're not going to go into any of the math or anything, just because there really isn't time. If you're in an open container, your solution has a pressure potential of 0, and if it was pure water with no solute in it, it would still have a potential of 0. As you add solute, you make the solute or the water potential more negative. And this relates to the different hypotonic, hypertonic, isotonic solutions, because water is going to go from an area of higher water potential to an area of lower water potential. You can see that here. More solute outside the cell, lower water potential, less solute inside the cell, higher water potential. So water is going to move from the inside of the cell, where there's higher water potential, to the outside of the cell, where there's less water potential. Same thing with plant cells. This formula is on your formula chart. Solute potential is always negative because you're decreasing the free energy of water. This is how you calculate solute potential. Once again, the formula is on your chart. Pressure potential is the physical pressure that's usually applied to a solution. You only have it if you're in an enclosed space like a plant cell, and if you're doing a problem like that, they'll tell you what the pressure potential is, or they will give you the information you need to solve for it. Turgor pressure is just the counteracting of the pressure of water inside of a cell pushing out against the cell wall. The Turgor pressure is that pressure pushing out against the cell wall, and the cell wall has to exert equal and opposite pressure. Things will tend to move until they reach equilibrium. So, for example, you have an initial condition, some potato cell stuck in some pure water. Things will move until they're at equilibrium and everything has the same water potential. So water will flow into that cell. Water transport in plants is essential. Water has to constantly move into a plant and then out of a plant. If you don't have that happen, then you can't transport nutrients to the leaves. So nutrients come in at the roots, they're carried by water, and they will move up to higher parts of the plant. So by adjusting its water potential, a plant cell can change whether water flows to it or away from it. And it can do that by dumping some ions in or some sort of solution. And that's just what you see here. So you have higher water potential, lower water potential. I know the numbers get bigger, but they're negative. So the bigger the negative number, the smaller the value. So water is going to always be moving up to the top of a plant and then out. And that process where it's leaving through the stomata of leaves is called transpiration or evapotranspiration, if you want to emphasize the fact that it evaporates. All right, let's talk physical structures of cells now. Organelles, the basic unit of structure and function. All cells have the following things. They have DNA, they have a cell membrane outside of themselves, they have ribosomes, they have a cytoskeleton, and they have cytoplasm. That's like the base model cell. Any extras that you add on would make you a different type of cell. So you can be either prokaryotic or eukaryotic. If you are prokaryotic, you're a bacterial cell. If you're a bacterial cell, you're prokaryotic. All prokaryotes are unicellular. There are no multicellular ones, so they're all microscopic. They have no nuclei. They don't have internal organelles. They don't have a lot of structure inside of them. Eukaryotes, if you can see it, it's a eukaryote. So animals, plants, fungi, and there's another group, the protists. I call them the trash heap of domain Eukarya. They're the things that don't fit anywhere else, so we just stuck them in kingdom Protista. But you can have things like algae, which are also eukaryotes and have chloroplasts, and they fit in over here because they also have nuclei. So prokaryotes have no nuclei. They have a nucleoid, which is just an area where their DNA hangs out. It's not separated from anything else. They're usually much smaller. Eukaryotes are much bigger. We have more DNA, more chromosomes, bigger space, more organelles. We're just more complicated. And those membrane-bound organelles, that compartmentalization, allows us to have different characteristics in different parts of the cell. So there you've got different organelles in an animal cell. When you're looking at this, when it's on YouTube, you can always stop and say, okay, let me pause and look at this. Those are other organelles in an animal cell. Here's a plant cell, and the rest of the plant cell's organelles. We're going to go through them one by one though, so that's why I skipped through. So ribosomes are translators. They make proteins. They're involved in protein synthesis. They are very super tiny. They have 2 subunits, so they kind of look like a mushroom or a dude with a hat and a mustache and nothing else on his face. I'm going to give Nick nightmares about that now. And they're made of ribosomal RNA and they're made of proteins. So they're a mix of 2 different types of biomolecules. They tend to float around in the cytoplasm, although depending on the protein they're making, they can attach to the endoplasmic reticulum, which makes it rough endoplasmic reticulum. And you can have a bunch of these reading the same mRNA, spitting out protein at the same time. So I talked about an endomembrane system. These things can all physically interchange bits of membrane with each other, and it's usually carried around or moved by vesicles. This is also important for the pathway along which we say proteins are made and packaged. So in the nucleus, that's where you have your mRNA made. That's transcription, and it's going to then send the mRNA out to the cytoplasm. This can be a ribosome that's attached to the rough endoplasmic reticulum. That's where you're going to make a protein from that mRNA. That's translation. Then, if it is stuck to the endoplasmic reticulum, the ribosome, that can be an integral membrane protein, something that's stuck in the membrane. Or you can have proteins build up in the ER, and then they just basically look like they're being pooped into the ER. And then those proteins can build up. You can send them on to the Golgi. The Golgi has a system of sacs, and you can send the proteins from one sac to another until it's ready, and then you send it to the cell membrane and export it. So that's the general pathway that proteins go. So this is just it in visual form. So in the nucleus, you send the mRNA out to the rough ER. Ribosomes are going to translate the mRNA, make proteins, which can then bud off and move to the Golgi. They can get passed from one sac of the Golgi to another, but eventually they're going to be exported to the cell membrane. So the nucleus, we know what it does. It is where you transcribe RNA. It is where you store DNA. It's also got this little guy in the center called the nucleolus or nucleolus, depending on how you want to say it. That's where you make ribosomal RNA and ribosomes. So they're made in the nucleus and then spat out into the cytoplasm. And it is physically continuous with the endoplasmic reticulum. So you can move directly from the nucleus into the endoplasmic reticulum. The ER, there are 2 types, rough and smooth. The major difference, as you can see, rough is bumpy because it has a lot of ribosomes. Smooth is not bumpy because it's got nothing attached. There are some other differences. They are structurally different. The rough ER usually is these flattened sacs. Smooth ER looks like a bunch of tubules, and they have different jobs. Rough ER is all about proteins. Smooth ER has nothing to do with proteins. So, smooth ER does a number of things depending on the cell type it's in. It can be used for storage, detox, it can be used to make lipids, it can be used to store calcium, but notice none of these have to do with proteins. Instead, if you look at the rough ER, it's protein. Well, that's not protein, but the membrane that comes off of the rough ER goes to the Golgi and eventually ends up in the cell membrane, and the transport vesicles are usually full of protein. And if I want to make the protein connection, usually the membrane that's produced has some protein stuck in it that were made in the ER. The Golgi, sometimes it's called Golgi body, Golgi apparatus, Golgi complex. That's why I just go with Golgi. It looks like a set of really melty pancakes stacked atop each other. It is a bunch of sacs of membrane that are right next to each other, one after the other. And like I said, it's got a lot of vesicles going back and forth between the different cisternae, as they're called. Those are the little sacs of membrane. And it also has vesicles coming in and vesicles leaving. It's got distinct regions. There is a side that always faces towards the endoplasmic reticulum, and the other side always faces towards the cell membrane. They're actually called the cis and trans region. Cis meaning facing the same part of the cell, part of the same cell. Trans meaning getting ready to transport. I'm facing the opposite, or facing what's going to exit the cell. Mitochondria, we've talked about several times, especially with cell respiration. Break down carbohydrates. This specifically has the electron transport chain and also performs the Krebs cycle. Remember, there's a part of cell respiration that happens out in the cytoplasm. Important parts are the fact that it has two membranes that allows it to build up a proton gradient. It also has some of its own DNA, some of its own ribosomes, makes a few of its own proteins, and that in mitochondria as well as chloroplasts is proof that they were, or is evidence that they were probably endocytosed by some ancestral cell, never got digested, and just happened to hang around for the next several billion years. All right, lysosomes. Sorry, I was still editing this. Lysosomes are little stomachs. They're vesicles, but they're filled with what are called hydrolytic enzymes. Those are digestive enzymes. Anything that gets put into a lysosome is going to be broken down. So if you're a single-celled organism, you can use lysosomes to digest your food like we use our stomachs. If you are a multicellular organism, you can use lysosomes to break down any unwanted proteins. You can even use it to break down any organelles that are old and breaking down and malfunctioning. You just pop them in a vesicle, mix it with the lysosome, and then they break it down to components that you can recycle and use elsewhere. So this is kind of how that happens. You usually have some other vacuole that's involved, and the lysosome is going to bind to it. These guys, by the way, are made by the Golgi. So they come from the Golgi, they go along their merry way, and they go and they digest stuff. A vacuole is designed for storage. There is a very large one in the center of plant cells, the central vacuole. You also have vacuoles in some protists that live in fresh water. Too much water is coming into their cells, so they have what's called a contractile vacuole that will squeeze it out. And then you have multiple functions for these guys in plants. They store pigments. They can also store poisonous things, so if an herbivore comes and eats the plant, when they break open those central vacuoles, when they break open the cells, they get something that either tastes nasty or poisons them. So it's a defensive mechanism. It gives you a little more surface area for your cell. You can store things in there. It creates turgor pressure because any extra water that's going into the plant cell goes in there. It can be used for reproduction to give flowers color, for example. You put pigments in the central vacuole, it makes it all pretty, attracts pollinators. And then you can also have some digestive enzymes in there when you have a seed, so that it can help break down the food that's stored in the seed when it's not photosynthesizing yet. Chloroplasts are found in algae and plants. They perform photosynthesis. They are very similar to the mitochondria in that they have multiple membranes. They have these little structures that look like coins called thylakoids. That's where, in those membranes, that's where the light reactions happen. Outside of that, you have a fluid called stroma, and that's where carbon fixation, aka the Calvin cycle, happens. And they have their own DNA, they have some ribosomes, they make some of their own proteins, and there's DNA evidence that they probably evolved from some cyanobacteria. Cell signaling. So, there are a couple of different types of cell signaling. If you look and you divide it all up, you can either have a response, excitatory stimulation, or you can have inhibitory signals that say, hey, don't do this function, or don't do this. So cells are constantly communicating with each other. Whether or not they're able to communicate with each other depends on whether or not they have a receptor. So this target cell has a receptor that can interact with this ligand. So it can have a response. This other cell, which may be nearby, has no receptors, and it doesn't hear the signal. It would be like, you know, with how the principals have walkie-talkies. These guys are all on the same channel. This guy's on a separate channel. He's not hearing anything. So, all organisms have to maintain homeostasis. If you are multicellular, you really need to be able to coordinate with other cells. That's why the cell signaling happens. If you are unicellular, then you need to be able to respond to your environment. Signaling molecules are pretty highly conserved across all the different types of organisms, which is some evidence that this probably evolved fairly early in life in prokaryotes, and then eukaryotes just adapted it for their own purposes. So, in single-celled creatures, you can use this to respond to your environment. So, a couple of different examples. So, you have quorum sensing, which we talked about. Bacteria will release a molecule, and when there are more bacteria building up, there will be more of that molecule in the environment. Once you hit a certain amount of signaling, they're receiving a signal from that molecule, and if they know the population density is too high, they'll either stop reproducing or they might change their behavior to now act as a group, and that's when they'll form a bacterial colony. Chemotaxis is behavioral. It's when an organism is trying to move toward or away from a stimulus. Positive chemotaxis is moving positively towards like a gradient of a chemical, and so bacteria can use a receptor that responds to a certain molecule to allow them to go towards that molecule. That's where they can get food or whatever resources they need. And yeast release these pheromones that allow them to, that will trigger their ability to melt into another yeast cell and mate with them. In multicellular creatures, this release of cell signaling chemicals is going to allow for different parts of the body to coordinate to do a specific task. So, for example, epinephrine is a molecule that when it's released widespread is going to trigger your cells to perform activities that will spit glucose out into the blood. So it will help increase your blood sugar. On the other hand, here we have, I know that's a complicated graph, but what it's mostly doing is it's showing how these chemicals are produced, estrogen and testosterone, which then will act on other organs in the body and cause an organism to potentially develop into a male or a female. So there's some temperature sensitive sex determination in reptiles, maybe some fish, and so they will have that environmental control of sex determination, and then their ovary or their testes can then release these signals to cause the organism to develop into a male or a female. All right, so cells can communicate directly when they have like tight junctions or when they have gap junctions or plasmodesmata, anything that allows cytoplasm to move between them. You can also have cells that are interacting with each other via surface molecules. This is common in the immune system. So this could be like an antibody, and this could be an antigen that's being presented on the surface of an antigen-presenting cell. You can also have local release. So paracrine signaling is when a cell will release stuff that's just supposed to be received by nearby cells. You can also have synaptic transmission. That is also very local. So these are two short distance signaling mechanisms. Plant immune responses are also kind of short distance. Like I said, they don't have immune cells that wander around their body. Instead, each cell has its own set of signaling mechanisms that, for example, will cause them to start making disease resistance proteins. And that's all the different proteins that are involved in the signaling pathway. You also have things like morphogens during development. These are things that help shape the body plan of an organism. So this cell will release it. This is paracrine signaling. The neighboring cells will receive the signal, and based on how much of the signal they're getting, they'll end up developing into certain types of tissue. So we looked at an example where, for example, if you were near the concentration of morphogen, you'd develop into like a thumb or a pinky, and if you were farther from it, you'd be the other type of digit, thumb or pinky. You can have long distance travel, so you have endocrine signals which use hormones that travel through the blood. You can also have hormones released in plants. They can travel through a plant's vascular system or directly from plant cell to plant cell through plasmodesmata. We have a number of glands that are in our body that release these hormones. They tend to have similar functions. So for example, things that are released by the thyroid gland tend to do with metabolism. Things that are released from the ovaries or the testes tend to have to do with secondary sex characteristics, and so on. So these are a number of the hormones that you have acting in your body. Big one is insulin. Insulin is released from cells in your pancreas, and when it's released, it will end up causing you to take up glucose from the blood and stop putting glucose in the blood. So it lowers your blood sugar. And if that goes wrong, then you end up with diabetes, which leads to increased blood sugar levels. Human growth hormone acts on muscles and bone. Here is estrogen acting on estrogen receptors inside of a cell. Here's testosterone acting on a testosterone receptor inside of a cell. And this is thyroid hormones, or some of the hormones released by the thyroid gland, and what effect they have on different organs. So you'll see it's like more fatty acids released, make more cholesterol, reabsorb cholesterol, make it into bile, make some fat. So there are various things that are happening when thyroid hormones are released. You can also have receptors that are on the inside of a cell. These are intracellular. Usually these are receptors for things that are hydrophobic and can pass easily through the cell membrane. So hormones like testosterone do that easily. So their receptors are inside of the cell, and then they can set off whatever sort of pathway they need to. That's where I wanted to go. A lot of times a receptor, the cell signaling is going to start when a receptor interacts with its ligand. It's very specific, so they'll bind to like one molecule type, and that will set off a series of changes inside of the cell. There are 3 different types of receptors. There's the G protein coupled receptors. They are what they bind to a G protein. The G protein, if it has a GDP, is not on. If it has a GTP, it is on and will make some other chemical, some other protein do its job. And then that will cause it to go back into the off state. And if you want to turn it on again, you're going to have another ligand come in and bind. You also have receptor tyrosine kinases. They need to act in pairs, so they form dimers when they are bound to their ligands. I believe insulin works on a tyrosine kinase receptor. And they phosphorylate each other, and then they can set off a bunch of signals. Ion channels, something binds, it opens it up so ions can flow in. Once that thing dissociates or comes off, the gate closes, no more ions can come in. Signal transduction is passing signals from outside of the cell to the inside of the cell, usually through second messengers. So this is the general signal transduction pathway. You have reception, where you get the signal, transduction, where it's being passed to the inside, and then finally response, which is whatever the cell's going to do. The signal transduction is usually carried out by second messengers. These are smaller molecules that diffuse through the cell, and they'll usually act on something else. A lot of them will end up being transcription factors or interact with other proteins. These are some of the major second messengers: cyclic AMP, calcium, and IP3, which stands for inositol triphosphate, though you don't need to know that. So cyclic AMP usually leads to the phosphorylation of proteins, so it turns them on. Calcium can act in another pathway that involves this guy, IP3, and then it can set off transcription factors that will then turn on genes. So here you see something like that happening. You have a G protein coupled receptor. It's got a G protein that gets turned on. It turns on this molecule, this protein, which is going to make IP3, which comes and opens up the calcium channel, and calcium comes out and does some other things. It's all super complicated. When you look at the diagrams of it, they look scary, but you should not have to know the details of any of these pathways. Most of what you'll need to know is this thing. And then finally, whatever your output is, there are 2 or 3 major outputs that you can have. You're going to either turn on a gene, so transcribe it, make some new product. You're going to induce apoptosis. You can say, okay, that's it for this cell, it's time to die. Or you can modify a protein, and there are 2 methods. You can either add some methyl groups, which can change its function, or you can phosphorylate it. Phosphorylation, oh, it's on another slide. Anyway, you can amplify a signal. Each step in the signaling process can go on and activate like 100 or 1,000 new molecules, so you can really build up a large response from a very small signal. Phosphorylation is adding a phosphate. A lot of times that gives energy to a protein, and it's like turning it on. And so you can add phosphates with kinases, which are a class of proteins that just add phosphates to other proteins, and you take them off with phosphatases. And when you either phosphorylate proteins or unphosphorylate them, you can turn them on or turn them off. So for example, this guy turns on this kinase, which takes this guy and turns him on, which takes this guy and turns him on, which activates this protein, which does a response. Most I would expect you to have to do with this is they would give you a diagram and you would have to interpret it. Yes, okay, so cell signaling is very specific, at least at the receptor level, but you can have multiple things happen once you get inside the cell. Depending on which receptor and which second messengers you have, you can do different responses. So that's why you can say have your blood release up and have your blood carry epinephrine and have it have different actions depending on the cell it goes to. So it can turn on some transcription in one cell type, it can turn it off in another cell type, because you have different signals on the inside. You can also organize your proteins in ways to make the signaling run faster. I don't see you needing that. Apoptosis is programmed cell death. It is usually triggered by some sort of cell signaling, and the cell will usually release like its own lysosomes to digest itself, or it'll form these weird things called blebs, which are a signal to white blood cells, eat me, I'm dying. And then you end communication by having the ligand come off, and this is a lot of times done to turn on or turn off gene expression. Oh, there's a lot of different things on here. I stuck like 6 or 7 things on here. So there's some things called cytokines. Their job is to help cells take on identities. This is developing seed. There's a hormone called gibberellin in them that when it's released is going to turn on or activate enzymes that are going to start breaking down the food that's in there, so that way it can grow. This is the pathway that a molecule called ethylene takes. It actually causes fruit to ripen. So what will happen is one apple on a tree will ripen, it'll release some ethylene. That ethylene will interact with all of these molecules and will turn on the ripening genes. So it'll make other fruits ripen. This is, so when you have this gene turned on, the SRY gene, that's what tells a body to become male or not to become male. So depending on the signals that are released, it'll either have your gonads develop into testes and you'll be a boy, or they'll develop into ovaries and you'll be a girl. All right,